So we said that infection is a fight of a pathogen and our immunity. We talk about pathogens a lot, and now we need to plunge into the concept of immunity. And if we can easily explain what is immunity, and I think that you all have some idea of immunity. So I understand immunity is the ability of the body to resist and protect us against all types of foreign bodies which trying to enter our body, including microorganisms, including toxic substances. If you have your own definition, it's fine, because immunity is too complex phenomenon to put the whole idea in one single sentence. So the basic property for us now is the ability to protect against diseases. Let's talk in that manner. Of course, we are not gods, and our immunity cannot protect us against every disease. And often we are affected by some disorders. And lack of immunity against certain disease is known as susceptibility. And person with no immunity against the disease is called not immune or susceptible to that disease. Of course, you all know we have a separate science to study immunity. It is immunology. And you could have another definition in the department. I understand immunology as the science dealing with the various phenomena of immunity, induced sensitivity and allergy. So we will give you only the basis, only theory of immunology. Detailed science, particular part will be provided on the immunology department in medical hospital. They will give you basic medical practice. So as you can see, the immunity is a unique system, and we extrapolate its properties on the whole body. We say the person is immune, and if immunity lacks, we say the person is sensitive. So the statement overall immunity is fine is known as immunocompetent status. And any immunocompetent person naturally is sensitive to certain diseases. Competent means all chains of immunity are working fine, and if any contact with the pathogen will occur, immunity will respond. Maybe we will be ill, but immunity will work in corresponding manner. And when that system is broken and person lose ability to respond, we're saying that person is immunocompromised. Immunocompromised host is a patient who does not have the ability to respond normally to an infection due to significantly impaired or weakened immune system. That state is more often temporary and can be caused by a number of conditions, including diseases, the same HIV, for example. Or it can be due to malnutrition, intake of some drugs, antibiotics or immunosuppressants, and some other. And if you will remove the reason of that suppression, you stop drug intake, or you gave correct treatment and you took HIV infection under control, that immunosuppression will gone. Your person will turn back to normal state. Person can become immunocompetent again. And as far as deficiency of immunity here is mediated by another factor, it's not immunological problem initially. We're saying that it is secondary immunodeficiency. You diagnosed a cancer, gave cytostatic drug to that subject to treat the cancer, and as a complication you've got an immunodeficiency in that subject. It is a second problem, that's why secondary. So constant immunodeficiency is more often related to some genetic disorder, to some chromosomal disorders, or some abnormalities in locus responsible for immune function. It can be structure of some immune cell. It can be structure of the organ. From the very beginning, the problem is the problem of immune system, those primary. And even we have very good significant progress in medicine, we cannot remove the genetic disorder still, and this problem is constant. And it's really unfortunately that we have no technologies. We have some ideas, maybe some hopes, but we cannot repair altered human genome yet. So you can see that problem is really complex, and as it usually happens, we have a complex approach to study that complex question. So we have multiple sites of immunity, and as a result we have multiple classifications of immunity in use. So the one we already started to discuss today, it is the origin of immunity. As we said, immunity it is initially originate from Latin innatus, means inborn, and means inherited by the organism from the parents. And immunity can be innate. Innate immunity protects us from the birth through the whole life. It is provided by different barriers, for example, skin. It is naturally given for everyone in the same quantity, in the same quality. And thus that immunity is often named as a natural. The main task of natural immunity is to prevent entry of any foreign agent into our body. And of course, mechanisms for that prevention are non-specific. Regardless of the nature of a foreign agent, the same mechanism of protection against chemicals, physicals, and etc. So acquired immunity is the immunity that individual acquires, of course, after the birth. And it will be induced by some factors that person will meet in the life. It is individual thing. It is reflecting the kind of adaptation of an organism to surrounding factors. So we said that it is adaptive. It will be induced by a particular substance, and immune response will be specific for that substance, unique mechanism. So if you already, I think, know, acquired immunity consists of specialized B and T lymphocytes and antibodies. Again, it is a topic of next lecture. Today the task is to understand first part, innate immunity. So as I mentioned, innate immunity consists of barriers, and we will have 5 types of barriers. We need to analyze every in details. So first one is obvious, skin. Skin is a mechanical barrier that prevents the entry of physical particles, chemicals, agents, and pathogens as well. Of course, mucous membranes. They can secrete some sticky mucus that traps microorganisms, immobilizes them. Microorganisms and dust particles can enter the respiratory tract with the air during breathing, but cilia of epithelium sweep the mucus loaded with the microorganisms and dust particles into the pharynx. From pharynx it can be swallowed or coughed out. It is a protective mechanism. We can activate coughing, sneezing, eye blinking, even vomiting is an attempt to help to remove pathogen from our body mechanically and not to allow them to invade. So biological barrier. We need to call to a memory. It is normal flora of human body. It is our protection. Besides all benefits we described already, it can prevent colonization of our body by pathogenic bacteria. Do you remember that? It's secreting some toxic substances, toxic for pathogens. We call it bacteriocins, specific antimicrobial proteins. Our normal flora competing with pathogenic bacteria for nutrients. It's competing for attachment to our tissues, and I promise you to discuss today biofilm formation. Biofilm is a very important trend in medicine now. A biofilm is an assemblage of surface-associated microbial cells that enclosed in extracellular polymeric some substance or matrix. Biofilm can be formed even by different types of microbes. Fungi, bacteria can stick together, fix to the tissues, secrete multiple mucoid substances, and make that kind of envelope to protect mechanically against physical factors, to protect against chemicals. It is a good barrier for other microbes. And it is a unique phenomenon because within biofilm, microbes can exchange signals. They can share plasmids. They can accumulate acid, bacteriocins to make chemical protection against pathogen. And that layer covers our intestine, our respiratory tracts, and giving protection against aggressive compounds of the air, of the food. Of course, it's not too much, but still it's something. It can even move. Center for Biofilm Engineering show that as minimum 5 types of movement of biofilms are possible. And the whole community of microbes can roll, seed, repel to change location. And that phenomenon is especially important in hospitals again, because microbes can expand bioplastic surfaces in the organs into other body. And the phenomenon of microbial communication within the biofilm, when the whole set of cells doing one business together, is known as quorum sensing. Again, it is a very important topic of multiple studies, and it was confirmed that microorganisms can share information on what happens around, and they can change their properties if needed. They can share plasmids to withstand action of antibiotics you will use. They can regulate density of the population to prevent infection or transmission of infection with the bacteriophages, and they can perform a lot of different things with it. It's really acting as a community, and it seems like microbes are much cleverer than the thought. And I don't believe I need to explain third type of barriers, is too obvious. But per contra, repeat action make perfection. So let's talk. Normal temperature of human body. 37 degrees Celsius is suitable for mesophilic bacteria, but it's not good for main number of microbes, making our body a Mars for thermophils and Sun for psychrophiles. Mesophilic bacteria are welcome, and they are feeling like in some health resort here, of course. So number 2, high acidity of stomach. Acid, it can kill main number of microbes which are ingested with the food. Bile, as you know, it's the factor which does not allow the growth of mainly gram-positive microorganisms, and that's why it is used in number of culture medium. Selective culture medium is the inhibitor factor. Airwax, it traps bacteria, dust particles, some insects, but excess amount is not very good for hearing. Remember that, please. Lysozyme, it is present in tissue fluids and all secretions except sweat, urine, and CSF, but it is present in good amount in tears and saliva. And lysozyme attacks bacteria, and it can dissolve cell wall. That's why some saying that lysozyme is an animal antibiotic. It's really so. So nasal hairs, it is a filter to filtrate microbes and dust in nose. They acting as a mechanical membrane, but as with the cerumen, excess amount not makes you looking really beautiful. Again, please remember that. So urine washes microbes from urethra. It is a mechanical factor. Vaginal secretions have some acidic pH, and it is not suitable for growth of multiple bacteria, and as well it can flush microbes out of the vagina. Sweat can make a protective acid film on the skin. It is inhibitory for many types of microbes. Tears contain lysozyme, mechanical washing, and ellipses, because I can forget even some factors, but if you remember, it is good. It's too simple to obvious, so moving forward. That is more interesting topic, and it is actually histology. And I'm repeating again and again, there is no individual science in medicine. All are related one to each other. Ilya Mechnikov, if you remember, in 1882 was studied response of animal tissue on bacteria and discovered immune cells. three science met together in one moment and gave a great discovery, phagocytic cells. So I really hope you studied well last year, because cells, it is the main type of mechanism of our protection. And even if to go on philosophical beliefs, it's all up to cells. We are made of cells. Bile, tears secreted by cells. Skin, it is a set of cells. And any type of protection is the cell mediated. But microbiology. So when we say in cell barrier, we mean, of course, immune cells. And immune cells which are involved in the fight with the pathogen. Number one is leukocyte. PMNL is polymorphonuclear leukocyte, very often PMNL, or neutrophils. They name so because they have, as you can see, multi-lobed nucleus. They're short-living, they're highly motile, they are phagocytic, and by nature they are killers. Neutrophils are formed from the stem cell in bone marrow, as you remember, I think, and they constitute the first line of the defense in the protection of the host from invading microorganisms. It's kind of obvious because they're small, they're highly motile, therefore they're much easier to get to the site of invasion than any other cell. So PMNL also contribute to the removal of the cellular debris from necrotic tissues during some reparative processes, during trauma, injury, or some other. And PMNLs are armed with highly efficient bactericidal mechanisms. Mainly we're talking about two. It is a phagocytosis, and they also can produce extracellular structures called neutrophil extracellular traps, or NETs. NETs are composed of nucleic acid of the cell and aggressive enzymes. Once triggered, the cell undergo a novel program leading to their death. While they perish, the cells release the content of their nuclei, and nucleic acid, DNA, mingled with bacterial enzymes, and that form that you can see it as a yellow. It is a lethal network outside the cell. So invading bacteria, pathogenic fungi, any pathogen get caught and killed here in the nets. That mechanism is very effective, but it is a suicide. Cell will lose DNA and lose nucleus. A lot of cells are required, and those neutrophils are the most numerous cells. They constitute 40 to 75% of all blood leukocytes in humans. So even normally they will die after a few days, and they must be constantly replaced. So second type of cell is a monocyte. They are the largest leukocytes. They are amoeboid in shape. They are present in bloodstream. Cytoplasm of that cell is clear without any granules, and those they are often named as agranulocytes. Nucleus, as you can see, is a bean-shaped. And monocytes, they constitute only about 2 to 10% of blood leukocytes. But they are motile, they are phagocytic, they can engulf bacteria, they can engulf cellular debris, and they can migrate from blood to an inflammatory site to perform their functions. So regularly, monocytes will circulate in the bloodstream only for about 8 to 10 hours. After that, they will start to grow, they will start to enlarge. And in 10 to 20 hours in blood, they will enter tissue spaces. And they will differentiate into specific macrophages, of course. Macrophage is a long-living and highly motile phagocytic cell. Of course, it is one of the most famous, one of the most known. Macrophages contain more cell organelles, especially lysosomes, and we divide macrophages into two main types. That one is a wandering macrophage. It has no fixed location. It shifts between different tissues by amoeboid movement. It has no stable location, no stable tissue. So fixed macrophages, they take up residence in a particular tissue. They serve different functions and the name to reflect their location. For example, where is it? That one, you can see osteoclast, bone macrophage. So that bluish with the violet nucleus cell, it is a osteoclast. So we have some microglial cells in brains, we have Kupffer cells in the liver, and we have some histiocytes in connective tissue, we have epithelial cells and some other cells, and you already discussed that one, for example. We already discussed, or we will discuss even functions of macrophages, so let's roll forward. Natural killers. NK cells are lymphocytes with the same family as T and B cells. They come in from a common progenitor. However, they are cells of innate immune system, means they do not have antigen receptors. Like T and B cells, and that's a difference. They are not specific to any antigen. They perform in fast response, even in the first encounter. And in the contrast to cytotoxic T cells, they don't need to be activated. So NK cells are present in spleen, lymph nodes, bone marrow. Regularly they constitute about 5 to 10% of peripheral blood lymphocytes in human. They can respond quickly to a wide variety of pathological changes in tissues. Best known for killing of cells infected with viruses. They can detect and control early signs of a cancer, as well as give us protection against diseases. And specialized NK cells are also found in placenta, and it seems like they can play some important role in the pregnancy even. So NK cells can cause cellular destruction in at least 2 ways. One important mechanism we will mention even later, it is a perforin production. Perforin is a specific system of proteins which can be inserted into the plasma cell of the membrane. They will create a pore in the plasma membrane, and they will allow entry of water into the target cells. Internal surrounding can get out, cell will burst, swell, and can be destroyed. Cellular debris, of course, will be cleared by phagocytes. So apoptosis, second mechanism, it is natural cell death or programmed cell death, which occurred as a part of a normal development. Normal maintenance and renewal of the cells, tissue, or organs. So one more important mechanism of cellular protection, it is a complement. Complement is a group of 20 proteins mainly produced by liver. Those proteins are present in blood serum and on plasma membranes and found circulating in the blood plasma within different tissues through the body. So they were named complement because they complementing the action of other compounds of the immune system. For example, action of antibody and antigen. So the main task of protein of complement is to prevent excess damage of host tissues, to destroy antigens, and we have 3 main mechanisms for that: cytolysis, inflammation, and phagocytosis. We will talk now. So complement is a cascade system. It means that in blood all compounds are inactive. They can have no activity, and the system needs to be activated in a particular order. Actually, you need to activate only the first protein of the complement, and it will activate next protein, next protein, next protein, and system will be self-activated step by step, thus cascade. So every further protein is activated by a previous one, and that mechanism, of course, is known and good described. So we said complement is a protective mechanism, and mainly to destroy antigens. So to activate complement, you need to have antigen. You already know that antigen should be bound by antibody. And of course, like here, like that. But no one will mention that actually to do that we need complement, because without complements antibody will not fix to antigen. It's not routinely said because it is too obvious and no one will mention that complement is required, but that's how it should look like. First, protein is involved in fixation of antigen to antibody, and when that complex is formed, complement will complete or compete function of antibody. Formation of that mechanism is the signal to start a cascade reaction. First compound will cleave second and fourth one, making C2A and C4B, and C2A and C4B together will make C3 convertase. As it's obvious from the name, that enzyme will convert third protein into two portions, and C3b will perform opsonization, do you remember, to help phagocytosis, as well as it will lead to formation of activation of mast cells and release of histamine. What histamine will do in tissues? Increase permeability of capillaries, diapedesis, attraction of new cells, leakage of different liquids in the tissues. Okay, C5b, it will activate sixth protein and attract C7, 8, and 9, and they will make that perforin system. They will make an open channel, balance of cell is damaged and cell die by cytolysis. That is a short scheme of regular complement activation, regular functions of complement, and that mechanism of activation is known as the classic way of activation. So as we said, one of the tasks of the complement is initiation of inflammation, and as you know, inflammation is a defensive response of the body to tissue damage. Inflammation can be initiated by pathogen, abrasions, some chemical irritations, different type of cell disturbances, and of course different types of inflammation are known. But regardless of the type, we have 4 common symptoms you should know by heart: redness, pain, local heat, and swelling. If you remember that, you can apply them to a particular area or organ and you will get symptom of the disease. It's easier. For example, tell me please, what is a phlegmon of a palm? Inflammation of adipose tissue in hand. Give me symptoms please. How to recognize phlegmon of the hand? So 4 symptoms you should know. Just take a look. We can see enlarged size of the palm. It is edema or swelling. Color is changed to red. If you will perform physical examination, if you will touch it, you will feel that it is really swelling and it will be hot. And of course, patient will feel pain when you will touch. In that picture, you can see that inflammation can lead even to loss of some function in injured area. If that person will have that hand, it will not play cricket for sure, because inflammation is an attempt to dispose of foreign material at the site of injury to prevent its spread to other tissues. If you will move, of course, microorganisms will move as well. So to localize that site of infection, to repair tissue and restore hemostasis. So in microbiology, we will talk, of course, about bacterial inflammation. We will talk about viral inflammation and partially about allergic inflammation. And they have some slight but some differences. For example, in allergy, pain will be transformed to itching. You all know it. Affected area is always scratched. And in microbial inflammation, you will have one more extra symptom. It is discharge. Complement will activate release of histamine, as we said. Release of histamine will attract phagocyte. And if it is a cell, bacteria or fungi, you will have pus, purulent discharge from the wound. In virus, discharge will have serous consistence because multiple cytokines will be secreted and other liquids. So one more important symptom is a fever. It can be induced by microbial toxins, or it can be induced by release of endogenous pyrogens by macrophages. So when enough amount of pyrogens will reach our brain centers, temperature of the whole body of entire body will rise. So we have some gradation of fever, and mild fever will activate phagocytosis and inhibit growth of the microbe. High fever is an attempt of our body to kill mesophilic organisms. If you remember, mesophils cannot tolerate temperature higher than 40 degrees Celsius. But of course, you need to understand that very high temperature is dangerous for our brain cells as well. And of course, high fever should be controlled. We need to control too high fever to prevent damage of our tissues. And here we have one common error. A lot of people take or given pills even for low-grade fever because it's bothering them. But you should remember that decreasing of fever will deactivate our protection. Remember that, please. We need to control only very high level. So cytokines. Cytokines are proteins which are released by our cells. They have specific effect on the interaction and communication between cells. Talking simply, cytokines, it is a language of our cells, and every cytokine is a unique code telling what to do, where to do, who should do. Literally, our immune cells is an army with different military branches. Every type of cell have unique options, and pathogen is an enemy. And during that fight, of course, our body need to lead the operation. It should be able to say to our cells something like, foot troops, sector B, fight please. Snipers, on the cover, artillery, sector A, ready fire. And of course, when enemy is defeated, everyone hold your fire. We need to control, we need to coordinate any type of cell-mediated response. And to perform that type of coordination, of course, we need complex language. And that language is language of cytokines. So we have cytokines according to a type of cell which is secreting. And we have lymphokines made by lymphocytes. We have monokines made by monocytes. We have interleukins. It is a cytokine made by one leukocyte and acting on another leukocyte. And we have chemokines. It is a cytokine with the chemotactic activity to induce chemotaxis. So cytokines have pleiotropy, means cells of different types secrete the same cytokine. Meanwhile, a single cytokine can act on several different types of cells. They can have different level of the effect. Cytokines can produce autocrine action. Auto means self-regulation. They act on the same type of cells that secretes them. They can give paracrine action. Paracrine means they will act only on nearby cells, those who are in close contact. Or they can produce endocrine activity. Endocrine means distant cells, far from the site of invasion. Cytokines are redundant in their activity, meaning similar functions can be stimulated by different cytokines. It is important if a production of some cytokine is affected or even blocked. We have kind of a backup. Another cytokine can perform the same functions. Similar to complement, cytokines are often activated in a cascade. And one cytokine will stimulate target cell to make additional cytokines. And you will have, again, the chain of reactions. And of course, system is self-regulated. Means that cytokines can have synergetic effect or they can act antagonistically. Where is it? When we need to activate higher some function, similar activity, cytokines will be produced and effect will rise. When task is completed, we need to reduce effect. Of course, to suppress already activated cells, not to waste powers. Antagonistic cytokines will be secreted and the state will turn back to normal. Our immune response will be suppressed. As you can guess, the main predominant producer of cytokine is a T cell, T helpers, and partially macrophages. So, in that scheme, in the diagram, it is a cytokine network. Schematic view of which cell can produce known cytokines and which cells are susceptible to those cytokines. And the scheme is really complex, as you can see. And of course, we will not ask you to remember all properties of every cytokine. No. But you should understand, and what's the key of that discussion, why I'm here. Maybe you know some factors already, some TNF or other, but system is too complex, and system is self-regulated. And amongst it, those factors, today interferons are the mainly important. So because they are used in therapy, and it is type of drug, but it was and it's still very doubtful topic, because very often it is not recognized as a drug. People, even some medical doctors, not realize that it is a potent drug with very serious effects and partially side effects, because it's looking like something we already have. It's harmless, means it's not a hormone like that. Why should it be dangerous? But it is a huge deception, because interference is the large family of alpha helical cytokines which are produced or induced in response to a specific extracellular biomolecule signal. And the name was given in 1957 because of ability of interferon to interfere replication of viruses. And by that effect, interferons are known widely. We have, as you can see, commercially available interferons, which are human interferons manufactured with the recombinant DNA technology. Mechanism of action of interferons is very complex, and it's not really well understood still. And it was shown that interferons do not kill cells directly. They do not kill virally or cancerous cells, but they can boost immune response to reduce the growth of the pathogen. And many viruses have devised mechanisms against antiviral function of interferons. They not only stop production of EFNs, but also they can disable proteins and some other molecules activated by interferons. So, as you can see, in current time, we have 4 classes and 3 types of interferons identified: alpha, beta, gamma, and lambda families. So, classes. Each class have many effects. Through any, some effects are overlaps. EFN1 belong to type 1, EFN alpha. It is produced by leukocyte infected with the virus. Beta interferons are produced by infected fibroblasts, belong to type 1 as well. Interferon gamma, produced by natural killers and T cells. And production is usually induced by stimulation of sensitized lymphocyte with an antigen, or with non-sensitized lymphocyte by mitogen, means chemical substance activating cell division, and it is belong to type 2. So lambda consists of 3 interferons, 28AB and interleukin 29, and it is third type of interferons. So interferons can stimulate extra and intercellular network to regulate innate and acquired immunity. It can provide resistance against viral infections. It gives us anti-tumor activity. They can enhance macrophages and natural killer cells, and that's why interferons widely used in the therapy of viral and cancer diseases, because it's looking like very strong and powerful drug. But they should not be used routinely. They should not be given for simple respiratory disorders, for example, because it is a cytokine. And as you already know, cytokines are pleiotropic. Besides those functions which are good and beneficial, interferons can have immunosuppressive activity. They can lead to some pathogenic changes in collagen diseases. For example, they are involved in the pathogenesis of diabetes, nephritis, and some other disorders. And of course, normally, signal for secretion of active cytokines will aware suppressive system as well. And if you are given some active cytokines externally, you activate it artificially, the system. Who will suppress it? Who will stop angry cells in the future? Actually, the same person should do who gave that cytokine. But of course, you will not give because we have nothing to suppress them. So please think, what are you doing before are you doing? And of course, save your knowledges to be able to think. I hope to see you soon. Have a good time. Bye-bye.